For decades, we've had the complete instruction manual for a human being, our DNA. The big problem? We could barely read it. But a revolutionary new AI is changing everything, turning our genetic blueprint from this mysterious, ancient script into a map we can finally actually understand.
Okay, so to put this in perspective, imagine you're trying to navigate a city with a map that's 3 billion letters long. But there are no labels, no street names, no landmarks, no "you are here" marker. That is exactly the challenge of the human genome. It's this incredibly detailed map of us, but without a key, it's just raw, overwhelming data. So for years, we've been trying to label this map kind of by hand, one tiny little section at a time. And let me tell you, it's been slow and honestly, often inaccurate. But what if? What if an AI could just look at that entire 3 billion letter sequence and fill in all the blanks? I mean, identifying every gene, every switch, every single crucial instruction all at once. Well, that's the breakthrough we're going to dive into.
All right, so to really get why this is such a huge deal, we first need to understand the fundamental challenge it's solving. How do you make sense of our own genetic code?
The scientific term for this is genome annotation. It's the process that takes that raw DNA data and starts labeling all the important features. So for example, it finds a gene, that's the recipe for a protein. Then it finds the promoter, which is basically the on switch for that gene. And maybe an enhancer, which is like a dimmer switch that can tweak its activity from really far away. Without these labels, the map is pretty much useless.
So how did we used to do this? Well, the old tools were incredibly specialized, and as we're about to see, that was their biggest weakness.
The old way was kind of like having a team of experts, where one person only knows how to find bakeries, another one only knows about parks, and the third, well, they only know post offices. They all worked separately, usually on these tiny little sections of the map, and they just couldn't see how it all connected. The new AI we're talking about, it's like one master cartographer who understands the entire city plan, labeling everything in context with incredible speed and accuracy.
And that brings us to the hero of our story, a new model that completely reframes how we even approach this problem.
Now, this quote here, it really gets to the heart of the innovation. You see, the researchers didn't just build a better tool, they changed the entire task. Instead of searching for individual things, they treated it as semantic segmentation. So think of it like this. Instead of trying to find every single tree in a satellite image, you just teach the AI to color in all the areas that are forest. It's this holistic approach, and believe me, it is a game changer for genomics.
So how does this new AI, called SegmentNT, actually work? Well, the concept is brilliantly simple. First, you start with a powerful foundation model that has already learned the basic language of DNA. Second, you train it to segment or, color in the different parts of that map. And third, and this is the amazing part, it does this for all the different types of features at the same time.
14, that's right. That's how many different types of genomic elements this one model can identify, all at once. We're talking protein-coding genes, those regulatory switches like promoters and enhancers, and even things called splice sites, which are the super precise cut and paste points for assembling genetic instructions. It gets the whole shebang, all in one go. It's just an incredible leap in versatility.
Okay, so a new tool sounds great on paper, right? But how does it actually perform when you put it to the test against the vast, complex landscape of the human genome? So the secret sauce here is something called pre-training. Before SegmentNT ever tried to label a single human gene, its core, the nucleotide transformer, had already been trained on just a massive amount of unlabeled DNA sequences. It's kind of like an expert who's read thousands of books on city planning before ever being asked to draw a map. It already gets the underlying patterns and rules.
And wow, this chart just lays it all out, doesn't it? On the left, you see the performance of the pre-trained model. On the right is an identical model that started from scratch. The pre-trained AI achieved more than double the accuracy. And not only that, it learned 7 times faster. This is some seriously powerful evidence for the value of foundation models in genomics.
So how does it stack up against the competition, the old guard? This chart shows a showdown between SegmentNT and Augustus, which is a really well-respected state-of-the-art tool. When you ask them to scan the whole genome, not just these little isolated gene regions, SegmentNT just significantly outperforms it. It's more precise, has better recall. It just shows its superior ability to navigate the entire map. And here's another huge advantage. Its field of view. Some genetic features are controlled by DNA sequences that are really far away.
Now, older tools could only look at a small neighborhood at a time, but SegmentNT can analyze a massive 50,000 letter long sequence at once. This ability to see the bigger picture is absolutely critical for understanding the long-range interactions that govern our biology. But this is where the story takes a really interesting turn, because the researchers didn't just build a better human mapmaker, they accidentally created something way more powerful. So that leads to the big question, right? This AI was trained only on the layout of the human city. Could it possibly understand the map of a mouse? A chicken? What about a plant? You'd probably expect the answer to be no.
And, well, you'd be kind of right. What's so fascinating here is the clear pattern. The model does a pretty good job on species closely related to us, like mice, but its accuracy drops the further away we get on the evolutionary tree. This tells us that while the fundamental rules of the genetic code are shared, every species has its own unique dialect.
So what did the researchers do? They created a multi-species model, fine-tuning the AI on the genomes of just a few other animals. The result was astounding. This new model became a true master cartographer. It can now generate highly accurate maps for species it had never even seen before, including plants whose genomes are structured really differently from ours. It had learned the universal language of life.
So let's zoom out. What does this all mean? An AI that can rapidly and accurately read the genome of pretty much any organism, that isn't just an academic achievement. It has profound real-world implications. So why does this new map matter so much? Well, with a better map, we can more easily spot the typos in our genetic code that cause disease, leading to faster diagnoses and new treatments. We can speed up drug discovery by understanding precisely how medicines interact with our genes. We can finally start to catalog the immense biodiversity of our planet by mapping the genomes of new species in days, not years. And, so critically, we can analyze the massive genetic rearrangements that happen in cancer, which could reveal new vulnerabilities we can attack. For the first time ever, we have a tool that can truly read the book of life, not just for humans, but across all species. The ability to instantly generate a detailed, functional map from just a raw DNA sequence is a power we have never had before. It opens up entire new worlds of discovery, and it leaves us with one really exciting question. Now that we can read the map of life, where's it going to lead us next?